Okay, Rakesh, I've launched the technical poll. Students, I've launched the technical poll. Quickly answer me through poll. My audio is clear and my screen is visible. You can see my screen. Okay, Rakesh, I've launched the technical poll. All right then. I know that in the first half you have your test, right? So how was it? Was it difficult? Was it easy? Was it moderate? How is it? Let me know in the chat box how you feel the test was. Because in the first half you have your test, right? How was it? Was it easy? Was it moderate? Was it difficult? What do you think? Rakesh, Mohina? Easy, very good. Okay, so in today's class, we are going to discuss the test, some question from the test. So it will be a paper discussion class. Okay. So let me proceed with the first question. Question number 1 says, which of the following is a sex-linked recessive disorder due to defect in either red or green cone of the eye, resulting in failure to discriminate between red and green color? Your options are A, night blindness, option B, color blindness, option C, myotonic dystrophy, option D is cystic fibrosis. So quickly answer in the chat box what you think would be the correct answer for this question. Sometimes the hints are given in the question itself. Try to find those hints. In this question itself, there's 2 hints. Students, try to find those hints. It's very important because if you're able to find these hints in the question, you can easily save your time in the exam. Okay? Let me know in the chat box. What do you think what would be the correct answer for this question? Which of the following is a sex-linked recessive disorder? Recessive disorder. Okay? Due to defect in either red or green cone of the eye. Red and green, what are they? They are colors, right? Resulting in failure to discriminate between red and green color. So quickly answer me in the chat box. Mohina, Ayushi, Trisha, what do you think? What will be the correct answer for this question? Okay, B. Ayushi says B. Mohina says B. Okay. Color blindness, Trisha says color blindness. Very good, everybody. Very good, very good. Okay. So, first hint is your sex-linked recessive disorder. So this is your hint number 1. Due to defect in either red or the green cone eye. This is your hint number 2 because they are asking about the color. Okay. And you can easily, from this hint, you can easily answer that it is color blindness. So your correct option is option number 2 or option number B, which is color blindness. But let me discuss with the option number 1, A, C, and D, why they are incorrect. Okay. So option number A, which is your night blindness. So night blindness, beta, it occurs due to the deficiency of the vitamin A. Okay. When you have the deficiency of vitamin A, this deficiency would lead to a disease which is your night blindness. Second is your correct option, which is a color blindness. And color blindness is a sex-linked recessive disorder. Okay, and this happens when the mutation occur in the gene which is present on the X chromosome, When the mutation occur in this gene, which is present on the X chromosome, that person is unable to distinguish between the red and the green color. So this type of disease, the color blindness, you can see more in males as compared to female, because males are heterogametic, right? They have one X and one Y. So one gamete will carry the X chromosome and one gamete will carry the Y chromosome. Because it is an X-linked recessive disorder, mutation is occurring on the gene which is present on the X chromosome. So male are more likely to get affected by this color blindness. Option C says myotonic dystrophy. Myotonic dystrophy is a disease where your muscles get weak. Weakness of muscles is due to a myotonic dystrophy, and it is an autosomal dominant. Autosomal dominant disorder. Okay. I told you the hint, that is X-linked recessive disorder. Myotonic dystrophy is an autosomal dominant disorder. Similarly, cystic fibrosis is an autosomal recessive disorder. In cystic fibrosis, what happens? The cells that produce sweat, that produce mucus or the digestive juice, they get affected. In cystic fibrosis, the main mechanism which get fail or which get interrupted or disturbed is the transport of the chloride ion. The failure of the transport of the chloride ion leads to cystic fibrosis. So the correct option for this question is color blindness. I hope this is clear. In detail, I've tried to explain this question, okay? Now let's move to the second one. Question number 2 says, how many different genotype and the phenotype are possible in ABO blood group typing in human beings respectively? They're asking about the ABO blood group. So what would be the answer for this question? A, B, C, D. What do you think? What will be the correct option for this question? They're asking about the genotype and the phenotype of the ABO blood group. Let me know in the chat box. Okay, everybody says A is the correct answer. A. Surya says C. Okay. Anybody? Ayushi says A. Very good, very good. Okay. Rishitya says A. Okay, very good. So mostly, like all of you have answered it correctly. That is A. But let me explain why it is A. It's asking about genotype. Surya, remember this, they're asking about the genotype and the phenotype respectively. That means they're asking about genotype first and then phenotype. Okay. So you know that ABO blood group is because of a multiple allism. Okay. You see this phenomenon in ABO blood group is multiple allism, where there's one gene, which is I. I is this one gene, and this one gene, they have 3 allele. That is A, B, and small I, we represent it with O, right? So let me just draw the genotype. For example, if you have the A, if the person is having an A blood group, it can have like 2 genotype. One, it could be homozygous, and one, it could be a heterozygous. Okay? If a person is having a B blood group, then it could be having like 2 genotypes. One is this homozygous, and other could be this. If a person is having an AB blood group, which is a case of co-dominant, because IA and IB both are dominant. Remember this, beta. If a student is if a person is having an AB blood group, that means it will have an IA and IB, both allele, right? And if the person is having an O blood group, small I, homozygous recessive, right? IA is dominant, IB is dominant, and the small I, it is recessive. Remember this. And we know that the dominant will produce the character or show its effect over the recessive one, right? So here also in this case, the IA is dominant, small I is recessive. So we can see that person will have a A blood group. So this person will have a A phenotype. This person will have a A phenotype. This person will have a B phenotype. This person will have a B phenotype. This person will have a AB phenotype, and this person will have a O phenotype. So genotype is 1, 2, 3, 4, 5, and 6. You're having a 6 genotype, but phenotype are 1A, 2B, 3 is AB, and 4 is O. The phenotype are 4. Right? 4 phenotype and 6 genotype. So the correct option for this would be A, because they're asking first genotype and then phenotype respectively. Respectively means that the first which is given in the question should be the first in the answer as well. So they're asking like genotype is 6, the first is 6, phenotype is 4, so this one is 4. So the correct option is A. Is that clear? Suraj, it is clear now? Okay, now we are proceeding with the next question. Question number 3. Which of the following feature is shown by both floral formula and floral diagram? Option A is estivation of sepal in floral bud. Option B is adhesion between parts of floral whorls. Option C is number of locules in ovary. Option D is arrangement of ovules within the ovary. So quickly, let me know in the chat box, what do you think, what will be the correct option for this question? They're asking about the feature which is visible or which can be shown in both floral formula and floral diagram. They're asking in both. What do you think? Quickly answer, beta, so that we can go fast. Okay, yes, we are running out of time. So let me do it very quickly. Option 1 says estivation of sepal in a floral bud. Estivation, you all know that the arrangement of sepal and petals, right? Arrangement of sepal with their unit, with their respective members with the sepal, and the arrangement of petal with their respective members like petals. Right? You see valvate estivation, there's imbricate estivation, there's vexillary estivation, right? So estivation of sepal in a floral bud. Where in floral diagram, this estivation of sepal is visible in floral diagram. Okay. In the diagram, you can see the arrangement of sepal, petals, but you can't see these arrangement, whether it's an imbricate, whether it's a valvate estivation. You can't see these estivation in the floral diagram, in the floral formula. Okay. Floral diagram, it is visible, but not in floral formula. In floral formula, you can't see the estivation of sepal. And question is asking is, which of the following floral features shown by both floral formula as well as floral diagram? Second says that adhesion between the parts of floral whorls. This is your correct option. Adhesion means that is the two different entities are attached together. For example, like Epipetalus, right? In which the stamens is attached to the petals. Adhesion, that is different, right? Where the two different entities are attached together. Where if I'm talking about the cohesion, that will be the between the similar units, like syngenesious stamens, monoadelphous stamens, right? Polyadelphous. These are your example of cohesion. But adhesion is between different entities like petals and stamens. Like stamens are attached to the petals. In adhesion between parts of floral whorls. So option B says adhesion between parts of floral whorl. This you can see in both floral diagram as well and in floral formula. And see in diagram, let me draw it roughly. This is a petal, 1, 2, 3, 4, 5. I've drawn 5 petals. I'm roughly drawing it. See, let's say these are stamens. And stamens are connected with the petal. If we have to show the epipetalous condition, we show it like this. Okay? And in floral formula, if I have to show adhesion, we show it like this. This is your corolla or the petal. Stem and endosperm, we represent it like this. And like this. Okay? We show that stamens are attached to the petals. So this is your correct option. The adhesion is present. The adhesion between the parts of the floral word can be visible in both floral formula as well as in floral diagram. Number of locules in an ovary. Number of locules. Okay, let me draw it. So this is an ovary and these are the ovules, right? So I'm drawing it very roughly. This is the placenta. And you can see these compartments, one compartment here, one compartment here. These are the locules which are dividing the ovary into 2 compartments. It could be a bilocular, it could be a monolocular, it could be a multilocular as well. But the number of locules you can see in floral diagram, but not in floral formula. Okay, so, and question is asking about both. So this is also incorrect. Option D says arrangement of ovule within the ovary. Okay, the arrangement of ovule, we call it as placentation, with this axile placentation, parietal, marginal, okay, basal placentation there. In floral diagram, you can see these placentation, the arrangement of the ovule as I show you here. Arrangement of the ovules in the ovary, you can see in this floral diagram, but not in the floral formula. Is it clear to everyone? So the correct option for this question is option number B, which is adhesion between the floral parts of floral, adhesion between the parts of floral whorls. So next question says the newly synthesized mRNA does not require any processing to become active. That is, there is no post-transcriptional processing in. Option A is protozoa, option B is bacteria, option C says anaerobes only, option D says all unicellular organisms. What do you think would be the correct answer for this question? Quickly answer, beta, so that we can move very fast. B. Tivenshi, answer B. Very good, Tivenshi. Quickly, everybody. Mohina, Ayushi, Trisha. Vijay says B. Good. Ayushi says B. Good. What about others? Quickly answer. Okay. So all of you have answered correctly. That is B. Very good, Tisha. So the newly synthesized mRNA does not require any processing to become active. There's no post-transcriptional processing. You know, we have studied 2 types of cells. Yesterday also I told you this. One is prokaryotic and the other one is eukaryotic. So in prokaryotic, the transcription takes place in the cytoplasm, okay, because they don't have a well-defined nucleus, they don't have the nuclear membrane. So transcription is taking place in the cytoplasm. Prokaryotes, they lack introns. Introns are absent in prokaryote. They don't have just junk DNA or the non-coding sequence. And because of that, there's no splicing mechanism. Splicing is absent in prokaryote that does not require any processing Once the mRNA is formed after the transcription, that mRNA is active, or that mRNA is sufficient, like functionally active to able to form the protein. It does not require any processing. So prokaryote, they're simpler, but eukaryote, they're complex, right? They have introns, they have coding and non-coding sequence in the DNA, right? And these, so once the mRNA is formed in eukaryote, it undergo processing, and we call it as post-transcriptional processing event. What are those events? Number one is your capping. The mRNA, when it is formed, it is immature, and we call it as heterogeneous nuclear RNA. So after processing, this mRNA become mature, and we call it as mature or the mRNA, right? And what are those processing event? One is capping, which is taking place at the 5' end of the mRNA. Second one is the tailing. Poly A tail is added at the 3' end of the mRNA. Third one is the splicing. As I told you that eukaryotes are complex, they have both coding and the non-coding DNA. So the removal of introns from the mRNA is the splicing mechanism. So splicing is present in eukaryote. That means the eukaryote, they show post-transcriptional events or post-transcriptional processing events. But in prokaryote, there's no post-transcription processing event. So number one is the protozoa. Protozoa is a eukaryote. This could not be an option. Second is correct, bacteria. As I told you, prokaryotes, they lack introns, the splicing mechanism absent. So there's no post-transcription processing event. Third says anaerobe. This option is very unclear. Okay, beta. So this could be incorrect. All unicellular organisms like prokaryotes are unicellular, but some eukaryotes are also unicellular. Like Chlamydomonas algae is a eukaryote, but it is also unicellular, right? So this option is also incorrect. I hope this is clear. Moving to the next one, how many plants in the list given below are the example of modification of stem? Modification of stem. Don't get confused with the modification of root or leaf. They're asking about the stem. Bougainvillea, euphorbia, rhizophora, turmeric, sweet potato, turnip, colocasia, opuntia, chrysanthemum. Option A, 3. Option B, 5. Option C, 6. Option D says 7. C is the correct answer. Okay, let me check. So first says Bougainvillea. I'm writing with initials so that we can save time. So B is your Bougainvillea, right? So what type of modification you see in Bougainvillea? That thorns. Stems are modified into thorns, and thorns are for defense mechanism from the herbivores. Second one is your euphorbia. In stems in euphorbia, they are cylindrical and fleshy. Okay, let me. So R is for rhizophora. These are your stem modification. Okay, and this one will be a root modification. All right. So rhizophora, you saw the nematophore roots. In rhizophora, the rhizophora grow in a place where there's swampy soil, marshy soil, and there's lack of oxygen. So in the search of the oxygen, the roots, they move out. If this is a rhizophora plant, okay, the roots, they will come out like this. And they have these pores. The pores are nematode, where the exchange of gases takes place, right? So it's a modification of root. Nematophores, they are a modification of root. Now turmeric. Turmeric is also a modification of stem, the underground stem. Okay? The underground stem to store food. Your sweet potato. Sweet potato is a modification of root. The adventitious roots, they become swollen to store food. Turnip. Turnip is also a modification of root. The tap root of turnip, modify or swollen to store food. Colocasia. Colocasia is a modification of stem. The stem, they get the solvent to store food, underground stem, okay? Opentia. Opentia is also a modification of stem, the phylloclade. Right? The stem become swollen and fleshy and perform the photosynthetic activity, and the leaves, they are present in the form of the leaf, they show the modification, which is a spine, right? Now chrysanthemum. Chrysanthemum shows, let me just draw it with CH. Chrysanthemum shows sucker. Sucker is a modification of stem. In chrysanthemum, what happens there? The roots, which are below, sorry, stem, which is the underground stem, the stem, let me just draw it. These are your underground stems. So stem move horizontally at some time, and then they move upward like this. This is what we call sucker. You can also see this in banana and pineapple. Okay. So 1, 2, 3, 4, 5, 6. So correct option is 6. And this rhizophora, sweet potato, turnip are the modification of root. So the correct option is C. Very good, beta. Whosoever answered this question. Now let's move to the next question, which says, which of the following is not an outbreeding device? Outbreeding, that means they're referring to cross-pollination. Or xenogamy. Self-incompatibility, it is a genetic mechanism. Okay? It's a genetic mechanism. C, very good, Ayushi. It's C. And it's Killingelai. I hope I'm pronouncing your name correctly. You answer the previous question, that's C. Very good. Okay, Trivenzi says C. Very good. Okay, so self-incompatibility is a genetic mechanism, okay, at which the self pollen or the pollen from the another flower of the same plant cannot germinate on the stigma of the same flower. Okay, for example, if this is a plant, and this plant, hypothetically, just imagine, has a genotype S1 and S2. Okay, there's a flower. Let me use the other ink. And this is a plant, another plant or a different plant, okay? They're not same. That is why I'm drawing it with a different color. And this plant is S3 and S4. Okay, these are the stigma. These are the pollens, okay? These are the pollens and the stigma. So if the pollen, this, okay, you see this plant is S1 and S2. So if the pollen of the same flower falls on the stigma of the same flower, this is the stigma. Okay, if the pollen of the same flower is fallen on the stigma of the same flower, in that case, this pollen will not be able to germinate. Because of this pollen tube will not form. Okay, the pollen will not be able to germinate on the stigma of the same flower because of rejection. Rejection could be in the style, rejection could be at the surface of the stigma itself because of this genetic mechanism. Okay, but if this pollen will fall on the stigma of a different flower. This is S3 and S4. This is the stigma of a plant which is S3 and S4. So this pollen will able to germinate. And they form the pollen tube. I hope this is self-incompatibility concept is clear. Now the second one says dioecious. Dioecious condition is, you can see in papaya. Papaya plant, they are unisexual beta. They have a male and the female plants are different. Okay, so dioecious is the condition where we can prevent self-pollination or the autogamy. Cleistogamy, when the flower does not open at all, that means the stigma and the pollen of the flower, they are present in a close proximity of each other. And the flower is not opening, that the only possible way for the plant to set seed is the self-pollination. And self-pollination leads to inbreeding depression. To avoid all these inbreeding depression, we prefer cross-pollination or the outbreeding. So in cleistogamy, flower is closed, right? So that flower will always undergo self-pollination. So this is a correct option because the question is saying which is not an outbreeding device. In which of the features, say the, okay, which of the following is not a outbreeding device? They are referring to cross-pollination. Option D says different position of the anther and the stigma in the same flower. This is also a feature of a cross-pollination if the receptivity or the adhescence of the anther are not synchronized, if the position of the anther and the stigma are different, if the size of the stigma and the anther is different, for example, like stigma is longer, anther are shorter, right? So these are some features because of which the flower can show cross-pollination. I hope this is clear. Moving to the next one. Next question says, select the incorrect statement. Okay, statement number 1 says in mature pollen grain, the vegetative cell is bigger and has abundant food reserve and a large irregularly shaped nucleus. Option B says multicarpillary Apocarpus gynoecium is found in Macleia. Option C says seeds are produced without fertilization in apomixis. Option D says seeds of black pepper are not perispermic. Quickly answer me in the chat box what you think, what will be the correct answer for this question. They're asking about the incorrect statement. Okay, some says A. Incorrect. Let me give the emphasis on incorrect. Okay, Trivanshi says D. Okay. Answer quickly so that we can move fast to the next one. Okay, D. MS Gauri Lakshmi says D. Okay, so those who have answered D, they're absolutely correct. Because option A says immature pollen grain, the vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. This statement is absolutely correct. You can see these pollen grain. This one is the vegetative cell. This one is the generative cell. Okay. The vegetative cell is bigger. In mature pollen grain, the vegetative cell is bigger. Yes, it is bigger. Has abundant food reserve. It is bigger in size, so it can store abundant food reserve. And large irregularly shaped nucleus. Yes, it has a irregularly shaped nucleus. So statement 1 is correct. Option B says multicarpillary Apocarpus gynoecium is found in Macleia. This is Macleia picture is given in NCERT also, student, where you can see like this. It is drawn in NCERT. See this carpels, multicarpillary like this. Right? You can see this diagram in NCERT. So multicarpillary Apocarpus gynoecium is found in Macleia. This is also correct. Seeds are produced without fertilization in apomixis. Absolutely correct. If the seeds are forming without fertilization, then the process is known as apomixis. D says seeds of black pepper are not perispermic. This is incorrect statement, but correct for the option, correct for the question, because question is asking select the incorrect statement. So the correct option would be D. Seeds of black pepper are not perispermic. They are perispermic, beta. They are perispermic. Perisperm means the persistent nucellus. When the nucellus is not degenerated and it remains persistent in the seed, that seed is known as a perispermic seed. And you can see this in black pepper or the beet plants. They show the perispermic seed because the nucellus is persistent. I hope this is clear. Let's move to the next one. Next question says, choose the correct statement about water potential. We have to choose the correct statement regarding water potential. Option 1 says the kinetic energy of water molecule is less if the greater concentration of the water is present in a system. They're asking about the correct statement. Okay. The kinetic energy of the water molecule is less if the concentration of the water is greater in a system. For example, if this is a water beaker and these are your water molecule. So your water molecule, the kinetic energy is the random motion of the water in the in a solution, I guess. Okay, somebody has answer. Okay. All right, student. So first option is incorrect. Why it is correct? Why it is incorrect? Because the kinetic energy of the water molecule is greater. Here it is written less. So the kinetic energy of the water molecule will be more if the greater the concentration of water is present in the system. If this beaker will have a lot of water molecule, kinetic energy will increase. It will not going to decrease. It will decrease when we are going to add the solute. Okay. Option B says the water potential increases if some solute are dissolved in pure water. No, it does not increase. It will decrease. Remember, this is this is a water molecule. If I add solute to this, the motion of the water molecule or the kinetic energy of the water molecule will decrease because of the presence of the solute. Okay, and the magnitude by which this the water potential is decreasing, that magnitude is known as a solute potential, and solute potential is always negative. We denote it by psi s, and it is always negative. Remember this. Option C says if a pressure greater than atmospheric pressure is applied to the solution, its water potential increases. Absolutely correct. If the pressure greater than the atmospheric pressure, if I apply the pressure to the solution, its water potential will automatically increase. Option D says the pressure potential is usually negative. No, it is usually positive. But in plants, positive pressure in the water column in the xylem developed due to transpiration. No, not positive, but a positive pressure you will observe in hydrostatic pressure. And negative during transpiration. Pressure potential would be negative during transpiration because of the tension in the water column in the xylem. So the correct option for this question is option number C. Let's move to the next one. Which of the following is true regarding the secondary construction present in chromosome? Okay, this is a chromosome. This is a centromere, your primary construction, right? And the primary construction, they have like disc-shaped structure. We call it as kinetochore, where your spindle fiber come and attach during the cell division. And here is the satellite, or we call it as secondary construction. Option A says it is a darkly stained part of the chromosome. No, it is not stained. It's a non-stained part of the chromosome. It is not darkly stained. Option B says this is present on few chromosomes. Absolutely correct. This is present on few chromosomes. Position of it is highly variable. No, position is constant. Option D says sometimes kinetochore is present on it. No, kinetochore is always present on centromere, which is your primary construction. So that spindle fiber come and attach. Okay, and let me tell you some few points. regarding satellite, the secondary construction, because you might get a question on this as well. The satellite, it also shows high degree of polymorphism. This I also told in the previous class, yesterday class. Shows high degree of polymorphism. Second, satellite, it contains non-coding DNA. Third, it contains repetitive DNA. Remember these points regarding satellite. If you get any question from the secondary construction, you will able to answer it. Moving to the next one. I think you might find this question difficult. But sometimes question length, according to the length of the question, some students think that it might be difficult one, it will be a tricky one, but no. Don't judge a question whether it's length. So question says choose the incorrect option for the given figure, and the figure is showing the rate on the y-axis is the rate of photosynthesis, on the x-axis the light intensity is given, and option A is the E is the light intensity where the maximum photosynthesis occur. Option B is the C factors other than light are limiting. Option C says A, the light is limiting. Okay. Option D says B, transition between optimum and the limiting value of light. What do you think? What would be the correct answer for this question? They're asking about the incorrect option for the given figure. Okay, so x-axis shows your rate of photosynthesis. Sorry, y-axis shows the rate of photosynthesis and x-axis shows the light intensity. Let me draw the graph here. This is your light intensity and this is the rate of photosynthesis. So if I increase the intensity of light, the rate of photosynthesis is also increasing. You can see this linear curve, linear line. It shows the straight relation between the rate of photosynthesis and the light intensity. That if I increase the light intensity, the intensity of the light, the rate of photosynthesis will also increase, right? But it will increase to a certain point. After that, if I keep on increasing the intensity of light, the rate of photosynthesis will not increase, or you will not show the higher rate of photosynthesis after a certain period of intensity of light. So this is the point where the light at which you can see the rate of photosynthesis is maximum. E is showing the rate of photosynthesis and D is showing the light intensity at which the rate of photosynthesis is maximum. Remember this, D is showing the light intensity, E is showing the rate of photosynthesis. Okay. This is A. So A represents that light is limiting. Okay, you remember the Blackman law of Blackman's law, the limiting law, right? Where the many factors are affecting a chemical process, then the factor which is minimal to its which is nearest to its minimal value. Remember this Blackman's law? The factor which is nearest to its minimal value will be the will be the limiting factor, and that factor will determine the rate of the reaction. If I change the magnitude of this factor, the rate of the reaction will also change. For example, in this case, the light intensity is low. It is minimal. But if I start increasing the light intensity, the rate of photosynthesis is also increases. But after this point, that is C, after this point, if I keep on increasing the light intensity, there is no effect on the rate of photosynthesis because the light is no more the limiting factor. The other factors become the limiting. They could be a carbon dioxide, it could be a temperature, it could be a water. Okay, the other factors has become limiting and light is no more the limiting factor. So option A says E, the light intensity where the maximum photosynthesis occur. This is a wrong statement. Because light, D represent, I told you that D represent the light intensity where the maximum photosynthesis occur. Option B says C factors. Factors other than light are limiting. Absolutely correct. Other factors becomes the limiting factors at this point. Now the light is no more the limiting factor. Now the other factors, they play the role and they have become limiting. So the factors other than light are limiting. True. Option C says A, light is limiting. True. In the starting, this A, the light is limiting. Light is the intensity of the light is very low, and when we increase the light intensity, the rate of photosynthesis automatically increase. Option D says B, the transition between optimum and the limiting value of light. Absolutely correct, because B shows the transition between the limiting and this is the optimum value of light. This is the value at which the rate of photosynthesis is maximum. So question is asking about choose the incorrect option for the given figure. So correct option is A. All right, I hope this is clear. Moving to the next one, read the following statement carefully. 1. Epiblast is found in grass embryo. 2. In typical angiospermic fertilized embryo sac, primary endosperm cell is triploid. Third says seeds of pea and groundnut are albuminous. Fourth is apple and strawberry are, for example, false fruit. Fifth says a recent record of 2000 years old viable seed is of Phoenix dactylifera. How many above statements are correct? Okay, so statement number 1 says epiblast is found in grass embryo. Absolutely correct. Epiblast is the rudimentary cotyledon or the second cotyledon. Grass embryo, they have a very large and a single cotyledon, which is the scutellum. Option 2 says in typical statement 2 says in typical angiospermic fertilized embryo sac, the primary endosperm cell is triploid. Absolutely correct. This is also a correct statement, because you remember double fertilization, the male gamete fuse with the egg cell and form a zygote, and then zygote form the embryo, and the second male gamete fuse with the central cell, right? They fuses with the central cell and they form a primary endosperm, and the primary endosperm has a triploid ploidy. Options, statement 3 says seeds of pea and groundnuts are albuminous. Albuminous means endospermous. Remember, seeds are of two types, the endospermous or non-endospermous, or albuminous or non-albuminous. So albuminous is the another term for the endospermous seed. So seeds of pea and groundnuts, they are not endospermous. This is a wrong statement. This is a wrong statement because this statement says the seeds of pea and groundnut are endospermous, but the seeds of pea and groundnut, they are non-endospermous. They do not have endosperm. These seeds are of dicot seeds, and dicot seeds are generally non-endospermous. They have that cotyledons which are very large, swollen, and fleshy to provide nourishment to the developing embryo. Right? Monocot seeds are endospermous seeds, but dicot seeds are non-endospermous seeds or non-albuminous seeds. Fourth number statement says apple and strawberry are example of false fruit. Absolutely correct. Apple and strawberry are example of false fruit. False fruit are those which are formed other than the ovary. Generally, the ovary on maturation it forming the fruit, but if the fruit is forming from the part of the flower which is other than the ovary, then it is result into a false fruit. For example, in apple and strawberry, the other part is the thalamus. The fruit is forming from the thalamus. Fifth statement says a recent record of 2000 years old viable seed is the Phoenix dactylifera. Absolutely correct, beta. Phoenix dactylifera, the seed of this plant is having a viability of 2000 years, and the seed of this plant is excavated from the Harold King's palace. Remember this point? Now, how many above statements are correct? So statement number 1 is correct, statement number 2 is correct, statement number 4 is correct, statement number 5 is correct, but statement number 3 is incorrect. So option C, that 4 statements are correct. Now, next question says the forelimbs of whale, bat, cheetah, human share similarity in patterns of bones. Forelimbs, hence in these animals, same structure develop along the different direction due to adaptation to different needs. This evolution is known as divergent evolution. This evolution is known as divergent evolution because the four limbs of whales, bat, cheetah, humans, they all have same origin but perform different functions. Okay. This is the characteristics of divergent evolution, and the structures are known as the homologous organs. Convergent evolution, when they have the different origin but perform the same function, like the wings of butterfly and the birds. They are having a different origin but they perform the same function, that is flying. And the structure are analogous. In divergent, they are homologous. Coevolution, when the two species, they are evolving simultaneously, like the plant and their pollinator. Sometimes the plant and if the plant has evolved simultaneously, the pollinator will also evolve. So it is an example of coevolution. Natural selection. Natural selection was proposed by Darwin. The Darwin finches. Remember the variation in the beak, where the organism adapt to the environment in order to survive, for the better survival. Next one says, question number 13, which says, which technique can be used to separate DNA fragments generated by the restriction endonuclease in a chemical reaction? They're asking about the technique which can separate the DNA fragments which are generated by the restriction endonuclease in a chemical reaction. Number one is the option says PCR, which is your polymerase chain reaction. Polymerase chain reaction, beta, it is involved in the amplification of the DNA. Ayushi answers C. Very good, Ayushi. The correct answer is gel electrophoresis. Very good, Ayushi. Option B says ELISA. ELISA is an enzyme-linked immunosorbent assay. And this ELISA is used to it is used to detect the antibodies. Gel electrophoresis, this is a correct answer. For example, this is a DNA. And these are the two sites where the restriction endonuclease cuts. Okay. So three fragments are formed. Number one, this. Second one is this. And the third one is this. And this is your agarose gel. And these are the wells. So if I load this DNA on the basis of their size, you can separate the DNA. The one which is smaller in size will migrate faster or will move faster in the gel. The one which are larger in size, they won't be able to move faster, so they will move slowly in the gel. Number D says pooling. Pooling, beta, is that when we do DNA extraction, generally the purified DNA, we extract that purified DNA so that it does not break with the help of the spooling trick. So when you go for further studies and when you pursue bio for your further studies, then you will have to do an experiment like extraction of DNA from any plant. Okay, then you will learn this technique, that's pooling. Next one is the hormone which act on melanocytes, melanin containing cells, and regulate the pigment of the skin. Number one is A, melanocyte stimulating hormone. B is your ACTH. Third is the growth hormone, and the fourth is prolactin. What do you think? What would be the correct option for this question? A. Very good, very good. Very good, very good. So the correct answer is A, melanocyte stimulating hormone. A is your adrenal. Option B is ACT, which is your adreno corticoid hormone. C is the growth hormone and D is the prolactin hormone. Prolactin hormone play a very important role in the development of the mammary glands. Growth hormone for the growth. If the growth hormone is secreted in a very high amount, then it will lead to a gigantism disease like Khali, right? And if the growth hormone they are secreted in a very low amount, then it will lead to a dwarfism. Moving to the next one, read the following statement about insulin and choose the option that correctly fill the blank, that is X and Y. So insulin is consist of two short polypeptide chain, that is chain A and chain B, that are linked together by the bond. You have to name the bond. And the proinsulin has an extra peptide called. You have to name that extra peptide. Let me draw quickly because we are running out of time. This is a peptide A. This is a peptide B. Okay, and they're linked with the bond, which is a disulfide bond. And this is the proinsulin, which is the immature insulin, right? After maturation, this is the B peptide. This one is the A peptide. And these are the disulfide bond. And this is the C peptide, which is cleaved or which is extra. So proinsulin has an extra peptide, which is the C peptide. And this A and B peptide, because insulin consists of short polypeptide, that is A and B, and the A and B are linked by the disulfide bond. So the correct option is option number C, which is X is disulfide and Y is C peptide. Okay, very good, Ayushi. Very good, Tivensi. Very good, Keningele. Okay, time is over, students. I hope this lecture is informative for you. Yes, Keningele, C is the correct answer. Very good.